Good morning, Year 12. Sorry that I couldn't be with you in class today. Hopefully you find this little video useful so we can continue to learn our new content and keep up to date with our course. So please have your learning booklets out and annotate the slides as we go through. I have done a couple of little updates as we go through here, so you might notice some extra images or things like that that you may need to take note of. So our technique for today is gel electrophoresis, and some of you may have used gel electrophoresis in previous courses and perhaps in Year 10 as well. But what gel electrophoresis is a technique used to separate proteins or fragments of DNA according to their size. We can think of the gel in gel electrophoresis as acting a bit like a maze. And when we add DNA fragments to that maze, we can separate them due to size because small pieces of DNA are able to travel more quickly through the gel compared to larger fragments of DNA. So we're going to have a little look to see how that happens today. One of the key pieces of equipment that we need to use during gel electrophoresis is what's known as a restriction enzyme. And a restriction enzyme is a special enzyme that's able to cut DNA at a special site. So that's known as a recognition sequence. And it's able to cut that piece of DNA to produce the fragments that are used in gel electrophoresis. So we're going to go through our steps of gel electrophoresis. Our very first step always that people forget on their exams is we need to extract the DNA from a source or get some DNA from an organism of interest. That's of course going to be a very small section of DNA, so often it will need to be amplified using PCR. And then we would use our restriction enzymes to cut that DNA into fragments. And those fragments will allow us to produce banding patterns that can then be analysed and compared to other organisms. So we've cut our DNA into little fragments, and so what we do is we place those fragments carefully into wells, which are like little holes at the negative end of a gel electrophoresis cell. Our DNA is actually negatively charged, which means, if you remember back to Year 9 physics, opposite charges attract. So the negative DNA is going to move towards the positive end of our gel electrophoresis cell. We add a bit of loading dye to our samples, and that loading dye enables the DNA to be visible. It's a fluorescent marker, so when we analyse our DNA bands, we're actually able to see what we have produced. When we turn on our electric current, that's when our DNA starts to move. And like I said before, our DNA fragments are negatively charged, so they move towards the positive electrode. This is what an electrophoresis cell looks like. A few years ago, there was actually a WACE question that used this exact image and asked students to identify what it was and what it was used for. It was quite poorly answered. Many students didn't actually know what it looks like. We are going to be doing a lab where you can do gel electrophoresis and we'll have a practice. But yes, this is the cell that we're going to be using. You can see the electrodes that are there and space in the center, which is where the gel will be produced. So that little square box, that's where our gel would sit, and that's what's going to allow our DNA fragments to move through. So we've added our DNA fragments to our gels, and we've turned on our power, and our DNA starts to move through the gel. The thing we need to remember about the gel itself is if we look at the little animation at the bottom, the gel's actually like a little matrix, and it's quite difficult for the DNA to be able to pass through the matrix. So if we've got small pieces of DNA, they're able to work their way through the matrix much more quickly, and they're able to get further away from that negative end and closer to the positive end of our gel electrophoresis cell. So due to that movement, we're able to separate our DNA into bands, and we can see a vertical gel electrophoresis cell. So we've got the wells where we'd place our DNA at the top. We can see that the larger molecules are closer to the wells at the end of the run because it's been harder for them to move through the gel. At the end of the run, we can see our smaller fragments have travelled further through the gel and they are closer to the positive end of our gel electrophoresis cell. And this is pretty typical of the banding pattern that is produced, and we can see that we have got our deer species. We would have 4 wells that would have been set up in this particular case with our DNA samples placed from each species into the well at the top. Once we turn on our gel electrophoresis, the DNA that has been cut into fragments is going to travel and separate out those fragments. So we can see in deer species number 1, who's our common ancestor, we do have some matches between that and species 2, 3 and 4. In our exams, you might be given a situation that looks like this where you would need to analyse the banding patterns and comment on whether or not an organism is related to other organisms that are found inside that well. So here's an example. This is an older one, so 2014 WACE. Basically, we've got an extended response style question, and it's broken up really nicely into what are the absolutely must-have pieces of information versus how we can go from a 7 out of 10 to our more A grade type student. So here it says that we've got, we need to analyse DNA from some different species of red and grey wolf and explain how gel electrophoresis could be used. So we've gone through our steps. So the first bit, we need to prepare our DNA, we need to cut it with restriction enzymes, we may need to amplify using PCR. Then our fragments are going to be loaded into gel, placed in the little wells that are in the gel. We turn on the electric current and the DNA fragments start to move. So because they're negatively charged, they will move to the positive electrode. Larger fragments travel more slowly, smaller fragments travel faster. So that's kind of the basic information that we would need for gel electrophoresis. Then to gain those extra 3 marks to make a really top quality response, we needed to, number 1, apply it back to the scenario. So remember our first slide, we said that we had to extract DNA from an organism of interest. In this case, it's our red and grey wolves. We've added a loading dye, so that would be one of the early steps just before we add it into the well of our gel electrophoresis kit. The gel's covered with a buffer solution, DNA is stained so it can be visualised, and the profiles are compared. DNA profiles allow us to look for differences also between individuals of organisms. And the way that they do that is they can analyse sections of our DNA which are unique to every single individual. These areas of unique DNA are what's known as STRs, short tandem repeats. And what a short tandem repeat is exactly how it sounds. It's a repeat of bases that occurs over and over. So we can see in person number 1, their repeat is GA, GA, GA. And that is repeated 3 times. Other people will have different amounts of repeats on their DNA. So we can see that person number 2 has got 4 repeats of the GA, and on their second chromosome, they've actually got 7 repeats. So when we run the gel, because 3 repeats is going to be less bases on our DNA, we can see that person number 1 has got a band that's travelled quite far, and then they've also got a band that has travelled not as far. The person number 2, they don't match up. Okay, they've both got different repeats, different length sections, so the bands are going to appear in different parts of the gel. One thing I want you to notice is down the left-hand side of the gel diagram, you'll notice that there's a scale. That scale is there to allow us to identify how many repeats are on that particular DNA, and it helps us to basically assign a size. So it's a known value, and by looking at this, we're able to determine exactly how many repeats each person would have. So if we talk about DNA profiling now, that is when we're using gel electrophoresis to find out differences of DNA in individuals. So at that individual level, comparing those STRs. Mentioned this already, so what are our STRs? They're our short tandem repeats. Usually STRs are 2 to 5 base pair sequence, so we had the GA repeat previously, and then we've got our variable nucleotide tandem repeats. These ones here are a little bit longer, but same thing. They are a repeat of the same nucleotides over and over. Both of these are found in our introns on our DNA. So you guys might remember that introns are our non-coding DNA that is spliced out during our protein synthesis. And this is basically what we see here. We can use this DNA now to show up differences in individuals. So if we're doing DNA profiling, we go through a similar kind of thing. We would need to isolate the DNA from cells. We carry out PCR to amplify the areas that we need, and then we would enter that into our gel. We need to extract the introns. So exactly like in protein synthesis, when we splice out the introns and get rid of that information, this time we're going to be using a special enzyme known as a restriction enzyme that's able to recognize these introns and actually cut them out of the DNA for us. Our restriction enzymes are basically molecular scissors. So they're designed to cut DNA at a particular marker point. So here we've got some uses of DNA profiling. Remember, when we're talking about any of our biotechnology, we need to know how, we need to know what, and we need to know why. So any of these, you could pick maybe 2 that you could use to explain when DNA profiling may be used. So a really good idea is to pick 2 that are going to be used for conservation, because we are in biology. So all of these ones are going to be quite useful for you. Here's a more recent exam. So this is the last exam where DNA profiling was actually asked for. So I would expect that we're probably due for another DNA profiling type question. So here we've got WACE 2020, really straightforward type question. They've given us a scenario, which is pretty typical for biology, and then list the main steps involved in producing that DNA profile. So only 4-mark question. I think we're probably coming up to a DNA profiling question that possibly could be more of an extended response again. Then we've got Otzi the Iceman. Some of you might recognise Otzi because I think in year 7 you actually do a little bit of a section of learning on Otzi. I'm not sure if it's in I don't think it's in science, but it's in a different part of the curriculum. But Otzi was a corpse that was discovered in the early 90s. And when they found Otzi, they found him very well preserved, considering that he had died about 5,000 years ago. So obviously, if we've got a specimen like this, we don't want to destroy poor Otzi. But we do want to actually analyse his DNA and see what that information can tell us. So we would extract a very small sample of DNA and then use PCR to amplify that DNA and make multiple copies so that we can then analyse it. And then we use gel electrophoresis to compare Otzi's DNA to that of modern humans. So they asked people around the world to actually donate a DNA sample to compare to Otzi to see if Otzi actually had any living relatives alive today. So they ended up getting about 1,200 people tested from all around the world. And of those 1,200 people, 10 people had a perfect match for his base sequence. So that would indicate that Otzi's actually got at least 10, probably a whole lot more direct descendants of Otzi alive today, which is pretty cool. One last thing, we've got WACE from 2013. How might we use DNA profiling to improve things like captive breeding programs? So by using our gel electrophoresis and DNA profiling, we're able to preserve the gene pool of our captive breeding organisms. Obviously, we want to make sure that variation is high if we are breeding organisms in captivity. Variation is the key for stopping extinction. So what we want to do is we want to sort of identify organisms that are closely related because we don't want to breed them together. So here's just another really nice little example of how we could use DNA profiling in the real world. Okay, people, I hope that you found that useful. Please just have a read of the information in your textbooks. Also, if you could jump onto your OneNote, the details are in Sector for that, and continue working through those activities. Have a really good day.